Hello everyone, my name is Glenn Schumacher. I am a master's student at the University of Southern Mississippi's Gulf Coast Research Laboratory, and for my thesis I am working with Dr. Kevin Dillon, Dr. Michael Andres, and Dr. Mark Peterson to explore the ecology of nekton along natural, altered, and living shorelines in coastal Mississippi. And today I'd like to talk about some of our early findings. So to start things off, coastal salt marshes are an important habitat type throughout the United States, particularly along the Atlantic coast and even more so in the northern Gulf of Mexico, where about 60% of the United States salt marsh by area occurs. Salt marshes also provide many ecosystem functions such as mitigating coastal erosion and eutrophication, providing habitat for a diversity of wildlife, and functioning as a nursery grounds for many commercially and recreationally important fishery species. Salt marshes also connect terrestrial inshore and offshore habitats and provide nutrients to other habitats via trophic relay. However, salt marshes are being continually lost and threatened by coastal development, where they are often replaced by shoreline hardening structures. And as such, managers have begun incentivizing the use of living shorelines, also known as alternative bulkhead designs or ABDs, which are designed to mitigate coastal erosion while maintaining natural elements. As you can see in the photo below of one of our sites, we have two strips of riprap to prevent some erosion and a strip of natural juncus in between. Now, work along the Atlantic coast has demonstrated that ABDs are capable of enhancing biodiversity and restoring some ecosystem functions, particularly those relating to chemical and nutrient cycling. However, there's a limited understanding of nekton use of ABDs, particularly in the northern Gulf of Mexico. Therefore, we will be monitoring nekton communities along ABDs, natural marshes, rip-wrapped or grade shorelines, and fully hardened bulkheaded shorelines to determine if nekton diversity, abundance, and trophic structure varies between these habitat types. The study is being conducted in Biloxi Bay, Mississippi. We have 5 sampling sites positioned along a salinity gradient. Our first 2 sites for this inland in blue is Ancient Oaks, and in orange is Camp Wilkes. During the last sampling event, these were classified by the Venice system as mesohaline with an average salinity of around 10. At each of these sites, we have an ABD station, a riprap, a hardened, and a natural station. Moving further towards the coast, we have D'Iberville in yellow and Fort Point in purple. These are both classified as polyhaline sites with an average salinity of around 18. And at these sites, we do not have an ABD station. We only have a hardened, riprap, and natural site, but they were chosen to complete the salinity gradient. Our furthest site along the mouth of the bay is Shearwater in light blue. This site is again classified as polyhaline with an average salinity of around 20. And here we have an ABD station, a riprap, a hardened, and a natural station. Nekton sampling occurred during the November-spring tide of fall 2020. Upon arrival at each station, we collected water temperature, salinity, pH, and dissolved oxygen with a YSI sonde, and then a beam plankton trawl was used to target larval fish, zooplankton, and benthic invertebrates. The BPL was 1 meter wide and fitted with 505 micron mesh and pulled for about 30.5 meters parallel to shore at a depth of about half a meter. Depth was then recorded at the center of the BPL so that a known volume of water was sampled. Immediately after, we deployed fyke nets to target juvenile and adult nekton. These were fitted with two 22-foot side wings and a 15-foot center lead constructed from quarter-inch mesh, and a 4-foot high by 6-foot wide by 6-foot deep center bag constructed from 1/8-inch mesh. Fykes were deployed at low tide and fished for a full tidal cycle, or approximately 24 hours, and all captured organisms were returned to the lab. At the lab, BPLs were sorted and organisms were enumerated and identified to the lowest practical level. The fyke catch was identified generally to species, enumerated, and weighed in bulk by species. We then took a random subsample of up to 20 individuals of each species at each station and individually measured and weighed them. From these same individuals, we collected and froze tissue samples for stable isotope analysis, and in the case of fish, collected stomachs for physical diet analysis. Stomachs were dissected and diet items were enumerated and identified to the lowest practical level, then grouped by operational taxonomic units. At each station, we calculated pooled gear richness as the total number of species encountered in both fykes and BPLs, and calculated gear and station-specific Shannon-Weaver diversity indices. We also calculated gear and station-specific catch per unit effort for select species, families, and operational taxonomic units, in BPLs as catch per meter cubed, and in Fikes as catch per hour. We also calculated the percent frequency of occurrence of diet items for species at habitat types as the proportion of stomachs a diet item occurred in. We tested for significant effects of salinity and habitat type on richness, diversity, and catch per unit effort using general linear models, and tested for relationships between habitat type and diet item composition using Fisher's exact test. We did find that richness was significantly lower at polyhaline sites. However, we don't have enough data yet to compare habitat types within their specific salinity regimes. Regardless, when you look at the effects of habitat type on richness, it's pretty clear that there is no strong relationship here. It is worth noting, however, that our highest richness of 35 occurred at one of our ABD stations, and our lowest at 12 occurred at one of our riprap stations. Similarly, we found that phyke diversity was also significantly lower at polyhaline stations. However, again, when you look at this at a habitat level, there is no strong relationship or effect of habitat type on diversity. I'll preface all catch per unit effort information by stating that we found no significant effect of habitat type or salinity on any of the groups we are about to look at. That being said, we did choose two operational taxonomic units that we believe are particularly important in the context of ABDs and natural marshes to look at further. The first being resident opportunistic invertebrate fishes, which are fishes that spend most of their life in the marsh and are exploiting edge habitat while they're feeding. As expected, the mean catch per unit effort designated by these black X's was much higher at natural stations than ABD, riprap, or hardened stations. If we break this apart and look at the driving families, Athronopsidae or the silversides is what was driving that extremely high catch per unit effort mean at natural stations. We had one net where we caught over 2,000 in one net, which really inflated the mean catch per unit effort. However, if we exclude these and look at fundulids, which are killifishes, and again a classic marsh resident fish, we again see that the mean catch per unit effort is highest at our natural site, again driven by an outlier, although not as strong as that above. However, the mean catch per unit effort at ABD sites is still noticeably higher than that of riprap or hardened sites, which suggests that ABDs may support near natural levels of abundance of resident marsh fishes. The next group we'll look at is transient fisheries decapod species, which consists of locally important commercial and recreationally fished decapods. Similar to our last group, we see the mean catch per unit effort is highest at natural sites, driven by a strong outlier, but the mean catch per unit effort at ABD sites is higher than riprap and hardened sites. This trend continues when we break this down into the driving groups. We see the same thing over here in Penaids, which is our white and brown shrimps, and for Calanectes sapidus, or the blue crab. So this suggests that ABDs can support near natural levels of abundance of important fisheries decapod species. We'll now look at the physical diets of resident opportunistic invertebrate fishes, which again consists of fish that spend most of their life in the marsh and are exploiting edge habitat when they feed, such as Athronopsids and fundulids. In the figure, we have diet items by frequency of occurrence. In blue, benthic amphipods. In orange, isopods. In gray, microplastics. In yellow, non-decapod crustaceans. In light blue, organic matter. In green, polychaetes. In dark blue, swimming amphipods. And in brown, terrestrial insects. A Fisher's exact test did find that there was a significant relationship between habitat type and diet item frequency of occurrence, and subsequent pairwise tests with adjusted p-values found that there was an especially significant difference lying between ABD and natural habitat types. When looking at standardized residuals between observed and expected diet item frequency of occurrence, we found that benthic amphipods, microplastics, and swimming amphipods were all unusually high at ABDs, while they did not occur at all at natural sites, which may be explaining this difference. And while not significant, it's worth noting that terrestrial insects, again in brown, occurred at ABD, natural, and riprap stations, but were never encountered in diets at hardened stations, which may suggest that ABD, natural, and riprap shorelines provide a better terrestrial linkage than hardened shorelines. Overall, we found no clear trends in richness or diversity. And although the relative abundance of some groups, such as marsh resident fishes and transient fisheries decapods, appears higher at ABDs and natural marshes, there was no strong trends here as well. Physical diets also remain unclear, and we recognize that their use is limited. They only provide a discrete time interval, and in the fyke nets there is the potential for net feeding. Therefore, we will continue to sample nekton in spring, summer, and fall of 2021, which will provide us with more complete diversity information that encompasses transient organisms that may only enter marsh habitats on a seasonal basis. We will also use stable isotope analysis to identify the trophic level and primary production source of important fishes encountered across habitats, and compare long-term trophic webs across habitats. We will use this information to inform stakeholders about the potential benefits of ABDs and inform management decisions and answer questions such as, can ABDs replicate natural marsh nekton communities, and can ABDs help support commercial and recreational fisheries? I would like to thank everyone at the USM GCRL and Mississippi Department of Marine Resources that has helped out thus far with the project, as well as our property owners for generously allowing us to set fyke nets and pull BPLs adjacent to their property. I would also like to thank our funding providers at the Mississippi Department of Marine Resources and the United States Environmental Protection Agency, and thank you to everyone for listening. And with that, I will be happy to take any questions.